It was
Okay,
good.
great. If it misbehaves, just let me. It often does. Let me know. Okay, great. Thanks for the invite. And the talk so far have been super insightful. And actually, they've lined up in a really good order. So I think that's the effort that Nadine and Mark put into into putting the program together. And as far as I know, none of the speakers have interacted with each other on this thing, so that the flow has been really good. But I'm going to kind of gradually move us in a slightly different direction and just think about what is blue carbon and really redefining what we consider a blue carbon ecosystem. And so we've heard a lot about carbon and a lot of inorganic carbon, some organic carbon, and the flow through the system itself. And this is maybe moving slightly towards Sergio's talk, and then also these longer term what we call storage. And so a bit about where I am. I'm based in at the Umeå University, which is in Sweden, and this is kind of what the view in the winter looks like from near my office. But we have this very unusual climate, which is it's -30 in winter and +30 in summer, so it's 60-degree range. And so it often gets me thinking about how these organisms can cope with these kind of extreme environments. But this is where we are. So this is Sweden here. This is we're up here, and yeah, we're about 40K from the main town, but it's quite a fun place to work. Okay, so uh, I won't gonna go too much into the details of what blue carbon is because I'm sure many of the viewers are aware, but one thing I do want to touch on just before I start, which I think Sergio did a good job of, is the terminology. And in the blue carbon and actually in the carbon world, there is a lot of confusion around the terminology of what different parts of the cycle are called. And the reason for that is that carbon biogeochemistry has been looked at for over 100, 120 years. And there's some papers, from the early 1900s, and the concept of blue carbon has been looked up for a very long time, but it just wasn't called blue carbon before. It was called carbon biogeochemistry. And then the different groups of scientists that look at all these different parts use different names to describe the different parts of the cycle. So I'll pick some. It might not be the ones other people use, but you'll get the idea of the part of the system I'm talking about. And so, for example, blue carbon was classed as a nature-based solution that has certain socioeconomic aspects to it. And now we've moved more towards a natural climate solution with co-benefits, also has some socioeconomic aspects to it. But we're talking about the process of drawing down carbon dioxide into organisms. And so the way this effectively works, and Sergio did a really good job of introducing it, that we're producing all these climate gases, carbon dioxide, and photosynthesize and create biomass by taking up CO2. So you're taking that inorganic CO2 and you're converting it to organic CO2 and locking it in an organism. That is called green carbon. And then we have also what's called blue carbon. And then blue carbon is a similar thing whereby we're producing all this CO2 and other climate gases that gets into the marine system in a variety of ways, either through the atmosphere or down rivers. And organisms take that up and they bury that carbon, and that's called blue carbon. And in the blue carbon world, much of the initial work in the last 20 years kind of mirrors what was done in forests and terrestrially and looked at not solely, but largely biomass creation. And so that's photosynthetic systems that photosynthesize and create biomass. And then what we've called the classical blue carbon systems are seagrass beds, salt marshes, and mangroves. So they're all photosynthetic organisms that are linked to the atmosphere in some kind of straightforward-ish way. Um, and so the questions that I'm going to try and think about today in my talk are, so is the paradigm of these classical autotrophic blue carbon systems still appropriate? now we know quite a bit more about how many of these systems operate. What is the source of organic carbon in these biogenic carbonate biotopes? What about calcification? It releases carbon dioxide, and we heard a lot about that yesterday. And also, can humans alter these blue carbon dynamics? And so I would argue for the last one, we know quite a lot about this, but it's just a bit of a kind of a mind bender for us. So, first question, and this is work that was a COVID project by one of my PhD students, Kelly. She did a great job. And so in this work, what we really were interested in doing is saying, okay, we've got a lot more information now. Let's look in the literature and try and find what evidence there is in the literature that systems maybe other than the classic three might be playing a role in carbon. I'm avoiding terminology for a moment, and you'll see why. Drawdown. What are the roots of that sequestration? Again, in quotes there, because different people use that term in different ways. What features might generate this blue carbon status in some of these ecosystems? And really, how does that then fall into our management and research priorities? And so, as a quick reminder, what about a classical? What we're referring to here are seagrass beds, mangroves, and salt marshes. And so those ones is I'm not going to talk about here in this initial bit. And so after going through all the literature, what we found was between this period, 2011, 2023, that really there was the papers that looked at other ecosystems fell into two categories. There were either those that were looking to see if an ecosystem was blue carbon, or we're looking to see if it was doing something else, but maybe it could then play a role in blue carbon. But the ecosystems really that came up were these seven. So mud and tidal flats, bivalve reefs, rocky reefs, polar zoobenthos, coral reefs, coralline algae, and fjord shelf sediments. And of course, three of those are what we would classify as classic calcifiers. And we found, yeah, that there's some evidence, not much in the literature. Some have got 50, 60 papers. And in all but really one of the cases, most of the work was probably doing other stuff, but then thought, this might be a blue carbon system. And then there was some follow-on work to see if it could be classified as blue carbon. But many of these papers, of course, this was in the infancy of what blue carbon is, were doing this in different ways. And so what we tried to do was look for features that were present in all these systems and all these research projects that might allow us to say, yay, this is blue carbon or not. And one of the key things that we had to think about was really what the root of sequestration is. And by sequestration, in this case, I mean very similar to Sergio's description. This is taking the inorganic carbon from the atmosphere and putting it in biomass. It is used in different ways in different groups, but then you need to get that biomass somewhere to stop it breaking down and the carbon that's in there be released back into the atmosphere. And that is burial. That is a separate process, which I'll come to in a moment. Or immobilization, I guess, as Sergio was saying. And in this case, it falls in two categories, and I guess the technical words are allochthonous and autochthonous. And so allochthonous is carbon that comes from somewhere else. It's not by photosynthesis, so it's not direct biomass creation. And I'll talk about autochthonous a bit later. But really, the way this might work is that you've got your CO2 that we've produced, and then maybe trees and forests create biomass, phytoplankton create biomass, macroalgae create biomass. These things then lose biomass. So either trees, leaves fall off trees, etc., phytoplankton sink to the seabed. And somehow they end up getting trapped and then buried in some of these other ecosystems. And some of these ecosystems might be good at trapping that external allochthonous carbon. Other ones might not be that good. And so what we ended up with is these four features really that cropped up all the time And what you'll find is that in most of these plots that I go through, or some systems just don't have any data. And that's because either people haven't looked at it or the system doesn't lend itself to those kind of measurements. But really, the systems the aspects that were coming up quite commonly were this balance between, which we've heard a lot about, carbon drawdown and carbon release. And that if we look here, the carbon balance per year, zero is, a net zero balance. But what we see is in many of these systems, look at coral reefs, even if we look at NEM, NCM, it spans across both balance, both release and both uptake. And that applies for other systems as well. Some systems, for example, it's maybe always positive, mudflats. Other places it spans across the zero mark. And photosynthesis is always above zero. It's biomass creation. Photosynthesis itself, though not net. And calcification generally is release, but not always. And we'll maybe talk about that in a moment. And so some systems do both, and we know that. I guess that's not new information, but it's interesting that the spread is quite large. The source of buried carbon is important. And what we've just split here is in two large categories. Was it marine or was it terrestrial? And what we can see for some systems that there's little information. Well, there was when we did this. I guess we did the survey 2.5 years ago. But where there are, you can see that in nearly all of them, if we look at the percentage contribution of the amount of organic carbon buried in the system, that it includes both marine and terrestrial, and it varies quite a bit between systems. Like mudflats, it's anything from 10 to 90% of either marine or terrestrial. Maybe in algal beds, it's a bit more marine. And then in sedimentary systems, again, it's a broad range. But the take-home message here is that you are getting terrestrial organic carbon buried in marine systems. That is really important. Burial efficiency, and so this is effectively, if you have your organic carbon, so a leaf or whatever, lands on the seabed, it isn't immobilized, the word Sergio used. It can still be broken down, and that carbon can go back into the system. And what you need to do is remove it from microbes. Now, you're never going to do that completely, but the faster you get it buried in the seabed, the faster it's in an area of low oxygen, the less likely that's going to happen. For most systems, there's barely any information on that, apart from sedimentary systems, where this has been the bread and butter of marine biogeochemists for decades. And what's interesting, you can see the values vary from terrible, it just remineralizes super fast, right through to when it's on the seabed, it ends up in the sea floor. And that is very site specific, and there's good reasons for why that might be, which is the next point, and that is whether these are depositional settings. So that means that's whether there is a large amount of other material coming into the system that then causes the organic carbon to then get buried. And now those values vary between systems, but one that I will point out here is that coral reefs, which are often thought to be low sediment accumulation places, so low depositional settings, parts of coral reefs have very high sediment accumulation rates. And those sediments are maybe coral sands, of course, but there are also other sediments, as you'll see. So if we compile all that information together, I won't go through this in exquisite detail, but we also include the features that you would class a classical blue carbon system against. So high primary productivity, high biomass creation, and high localized sediment accumulation rate. We end up with a bunch of ticks, crosses, and maybes. And we look at the balance of all those. So is there a preponderance of maybes? Is there a preponderance of no's? At present, there's a few question marks, but the ones that we have a reasonably confident interpretation of that could be called a blue carbon system although maybe the biomass creation is external, are mudflower needs some coral reefs, not the whole reef, but the lagoons are quite important. We heard a lot about the other parts earlier on, coral algal systems and sedimentary systems. And so what really is the processes that are going on in these systems that facilitate them being classified as blue carbon is that they have this photosynthetic calcification carbon balance. Talk about a bit more on that in a bit. They have high rates of allochthonous carbon supply. And why this is really important is because that is organic carbon that may have been classed as a loss from terrestrial systems. So it was just classed as gone, but actually it's just been transferred into the marine environment and then buried in the marine environment. Maybe not the full amount, but that is an important addition. You have low rates of sediment carbon mineralization, so they're buried and then it's stored. You'll get some loss and you get loss over time. And these are highly depositional settings in many cases. And so where you have those features present in these, what we class as non-classical ecosystems, we can probably be quite confident that we can classify them as a blue carbon ecosystem. And so really this led us to produce a framework that one might consider. It follows on from some terrestrial frameworks, in fact, there's REDD+, etc., whereby you might think, okay, maybe we should be, managing this system because it's a blue carbon system. Now, I'm not going to go through all these boxes individually. There are key issues here that, of course, have been discussed previously, like we need to think about socio-ecological factors, etc. But one key thing, which is where the social aspect comes in, that you need to be able to manage the system for those features. Yeah, they've got to be something quantifiable. And this is where the problem comes in at present because of the degree of knowledge. And you saw all the stars on those figures. And one example, which is probably one of the best researched examples in the world, are coral reefs. And one of the things that we looked at, and I know there's a few people in the audience that are on this paper, I know Buki's in the audience, is that we really got together, and this was a lot of the heavy lifting was done by Heidi and Tess here, and looked at how much we actually know about these physical factors that are important in the way coral reefs survive or grow, and are they used in management and restoration of reefs. The results are surprising. So we looked at 157 papers, and of those 157, 55 peer-reviewed papers that were looking at either restoration or management of coral reefs didn't use any environmental parameters in the decision-making process. 55%, it's a very high number, often because the data wasn't available. And then if we look at some of the values that were in places where they did include, and some of the things we talked about the last day and day and a half, carbonate chemistry is only present in 3%, and geomorphology is only present in 5%, hydrodynamics only present in 6%. And this is for, a global, I guess, distribution of coral reefs. So, and this isn't a criticism of those papers, it's that often those data are not available. So trying to manage in these systems in the context of them being a blue carbon system is incredibly difficult because the graspable data often don't exist. And so this leads us to, even though blue carbon is thought to be reasonably well understood, there are a lot of unknowns, especially for these classical systems. And I'm not going to go through them all, but one that was touched on earlier by Isaac, and I'll touch on in a minute, it's not just about the carbon dioxide. There are other climate gases which we also need to resolve in these systems and consider them as CO2 equivalents if we are going to start managing these systems in the context of their role as a blue carbon repository. And I think with these non-classicals, we're definitely not there yet. Got a much better understanding for the classicals in that context. So I think that's something that hopefully will develop more over the coming years. Okay, so it is the paradigm of the only the classicals acting as blue carbon is still appropriate? I think the answer is probably not. Other systems bury a lot of organic carbon. It's maybe they just don't create it on site, but they do hold it. And we should probably have a think about how we manage those. And critically, we need to think about where that carbon came from. If a system is burying loads of organic carbon that isn't its own, but we're not managing the transit route of that carbon, it's pointless because that carbon might end up somewhere else if someone destroys the river, for example, etc. Okay, so what's the source of that organic carbon? Now, we've touched on that already, but just to unpack a bit, and I'll use chronalgal beds and deposits as an example here. I'm not going to go into it too much because I think Nadine's going to expand tomorrow. But these are calcifying ecosystems that have other organisms present, but they're not solid often. Sometimes they are, but they trap a lot of organic carbon, which they bury below their skeletons. And what we looked at is, for example, how much carbon is buried in these systems. And this is one of my PhD students, Jinhua, who did much of this work. And for temperate systems, at least, if we look at the top 25 centimeters of the system, the amount of organic carbon present is not that different to seagrass beds, marine sediments, or kelp forests. And so these are comparative systems. They're not small players in the total amount game. But what is important to show here that both Kelly and Jinhua have looked at this is that it does vary quite dramatically from site to site. It's not consistent. And often, if we look here in Scotland at 3 sites, that is to do whether the site is sheltered or exposed. And really what happens is when they're exposed, so this is a quite pixelated image, I'm afraid, of a seabed with these calcifying algae on, you get these mega ripples forming, and basically the waves turn up the sediment and reoxygenate any carbon, organic carbon that's buried, and expose it to being mineralized. And so this exposure has a big influence, I should say, on how much carbon is buried. And so really what Jinhua did, so back to Jinhua's original study here, is I try to identify or fingerprint where the carbon came from. And we can do this with stable isotopes. It's done a lot in terrestrial systems. And in abstract, briefly, you can look at the isotopic carbon and nitrogen composition of your organic material, and then you take what we call N members, but basically they're standards. So you take some marine macroalgae, etc., so you know what the fingerprint is. And then the colored dots here are the isotopic composition of the organic carbon material containing the organic carbon that's buried in the sediments. And what we see is they don't all fall. So this group of the standards, I guess, are the marine ones, and these are the terrestrial ones, but sometimes buried in the marine environment, and they don't fall purely in the isotope space of the marine fingerprints. They cross both, and they include marine macroalgae, but they also drag down in this direction down towards the terrestrial plants. Now, they are color-coded, so in this case, some of them are 5,000 years old. So the organic material was 5,000 years old that was buried in these systems. So this isn't just young carbon, it's old carbon, some of it. I won't unpack that too much in the context of today, but just to say that what we've got here are 2 terrestrial types of carbon, oak marine and oak terrestrial. What these are oak leaves from the tree and oak leaves that are on the seabed. And the important thing is their isotopic composition doesn't differ. So that means we can reliably use isotopic composition of most organic materials that have been dead for some time as a reliable fingerprint of what they look like when they're alive. So take-home message is that we have lots of different sources that are buried in the seabed, some of which are terrestrial. Now, I want to touch on the fleshy seaweed conundrum. And I know Sergio did this. And really, the take-home message here is, as Sergio said, seaweed is generally pretty short-lived, okay? It's just a few years. And you will have seen a lot of press and a lot of papers recently that that seaweed are going to be a key contributor to the blue carbon story. And really, this is the theory of how it works. So you've got your seaweed, yeah. They're doing all sorts of stuff. They're taking up CO2, they're releasing CO2, they're releasing methane, and they're releasing various nitrous oxides of nitrogen. But the balance across those probably means a sequestration of carbon or a drawdown if you use these as CO2 equivalents. So CO2 drawdown. This seaweed dies, and then it starts to break down. And then for this process to work, you need to get these bits of seaweed before they rot to be buried somewhere, presumably the seabed, to stop them rotting. Because if they rot, microbial activity, organic carbon is remineralized back into inorganic carbon, back into the system. But much of the stuff that we hear and see is only this part of the story. It doesn't include this bit of the story. And actually, we know very little about this bit of the story. And I think, as you saw in the previous figures, macroalgae does appear as one of the sources that we find buried in the seabed. But how reliable is that? And so I'm missing a slide, but it's okay. So, but what Jinhua and Kelly did was they went to a bunch of fjords on the west coast of Scotland, cored these fjords, and then looked at the isotopic composition of the organic carbon that is buried in those fjords to try and identify where the organic carbon was coming from. These are near to shore, so you should get a reasonable signal. And they found that, so these are isotope mixing model plots. Don't need to worry about too much how we get them, other than the peaks represent the proportion contribution by source. I apologize about the colors. Green is not algae. It's not photosynthetic. It's actually marine fauna. But the one we're looking for here is the brown one, on my computer at least. And what we found is for 3 of the fjords, the brown peak is down here. We're looking at it's only contributing 4 or 5% of the total mass of organic carbon buried in the system. But for 2 fjords out of the 5, it's about 25%. And so the take-home message here is that it is inconsistent. So sometimes you get a lot of the seaweed making it effectively into the seabed, so it's buried. Other times you don't, which means probably it's remineralized back into the atmosphere. So 2, what is the source of organic carbon? So it's both terrestrial and marine, and therefore we need to consider the delivery pathway. So the next 2 points are much briefer, so hopefully I'll get to them quite quickly. But what about calcification? We've heard about this quite a lot. It does release carbon dioxide. And I'm not going to go too much into detail. We've heard this all, especially there was a lot yesterday about this, but at geologic timescales, the inorganic carbon is effectively locked in some kind of structure, and it's not in the atmosphere, Great Barrier Reef. And at calcification timescales, so minutes to seconds, the carbon dioxide is produced. And we saw many of these plots yesterday, which are important. And I've got carbon dioxide in quotations there because it's not always as carbon dioxide, depends on the biomineralizer and how it biomineralizes. And actually, for many biomineralizers, we don't know fully how they biomineralize. But really, what we're now talking about is autochthonous CO2 uptake. So this is the organism photosynthesizing and taking up CO2. So they take up CO2 and then they calcify and they release CO2. And Sophie Martin, 2007, did a lot of this work and did some great stuff at the infancy of understanding how this works. And she found that, for example, for calcifying algae, we have in some cases, a release of around 40 grams per meter squared per year because of calcification. And that's great. That gives us a really good baseline to work from. And then since then, there's been a lot more work done on biomineralization and how it works. And really what we were interested to look at coralline algae as individuals and say, well, are they reusing some of this CO2 that they're producing by calcification as biomass, for example. And so, without going into exquisite detail, we use the dual radioisotope tracing incubations with carbon and calcium. And again, sorry, Xinhua drove this quite heavily. I don't think he slept for several months while he was doing the experiments. So what we were looking at is where does the carbon and the calcium end up, and what was the pathway? And we did this using both these tracers, but also using carbonic anhydrase inhibitors. And really what we found, it's more complex. I won't go into the full complexity of it, is that you have photosynthesis taking up CO2, but you have a reasonable degree of internal recycling of the CO2 that is produced by calcification, around 25%. And that ends up either in biomass or as part of the skeleton. And really, that means that if you do the maths, 40% of the calcification produced CO2 was not released. And so it was recycled within the system. And if we think about that, that means, of course, they are still releasing some carbon, but not as much as we initially thought. And really, that means that if we then start to think about, and we're not there yet, but how we fold the inorganic and the organic carbon sink together, if it's the inorganic component that wasn't released, we're looking at some reasonably large numbers of tons per year for corallines, but these are a gross underestimate. We don't know what the distribution is to start off with, and we don't know how deep, thick most of the beds are. But of the released CO2, what's important is these don't occur as individuals. They occur in ecosystems. And we know already from actually Sophie's work that I previously showed, but also from some of Heidi's work, that many of these systems are net autotrophic and they have a net drawdown of CO2. Yeah. And I think actually some of Nadine's recent stuff has shown this as well. And so even though they're releasing CO2, not as much as we initially thought, other components of the same ecosystem are probably taking up some of that CO2. So yes, they release CO2, they're calcifiers, and there's no way, there's no way of saying they don't, I think, but it's not as much as we previously thought because there is a high degree of recycling. And very quickly, my last point is a bit left field, but I think it's important, is that I think we have a reasonable understanding about how humans can interfere with this blue carbon dynamics in these calcifying systems, habitat destruction, climate change, changing transit routes, organic carbon. But ecosystem cascades in these systems are incredibly important. And this is some of the work we did with Doug a few years ago, looking at if you mess with the ecosystem cascade, so something completely different to what you think the carbon is being used for, does it mess with the carbon in that system? And one of these kind of unique, or I guess charismatic ecosystems, are the Aleutian Islands in Alaska. And you have this system which is driven by sea otters, basically. And so sea otters like to eat urchins, and then the urchins live on the seabed, and urchins will only eat coral and algae as a last resort. They much prefer kelp. And so kelp, as we know, sequester a lot of carbon. And while coral and algae, as we've seen, maybe don't emit as much CO2 as an individual, as we previously thought, it's generally lower sequestration. And so you have this system that's dominated by sea otters that eat the urchins, and therefore the sea otters are controlling effectively this flip-flopping between whether a system is in a lower sequestration state or a higher sequestration state, assuming a large degree of this kelp ends up buried, of course. And what we did was we actually looked at the growth bands that are locked within the calcifying algae and looked at grazer marks to determine historically what the relationship had been between otters grazing on the sea urchins grazing on the seabed, and then the flip-flopping between a high sequestration, low sequestration system. And what we found was that if we look at 4 sites, these are the grazing events per year. So up here you have a lot of sea urchins, which means this bit of the system, low kelp. Down here you have fewer sea urchins, which means less grazing, more kelp. And then something weird happened in the 1990s, which meant systems went from low grazing state to a high grazing state. And this something weird was actually hunting and disease. So by changing hunting patterns on sea otters, this messed with the sea urchins grazing on the fleshy macroalgae. And what this meant is this went from a system that was low grazing, high sequestration to a system that was high grazing, low sequestration by changing a law or the regulations around not only, because it was disease as well, the hunting of sea otters. So ecosystem cascades are really important, even in these strange ways that we don't think about. Okay, I will summarize it there. So non-classical systems bury a lot of organic carbon, probably not their own. And so we need to consider the transit routes of that organic carbon to the system. And humans can interfere in these systems in all sorts of weird ways, not always wonderful. And I think any management framework that we put in place around that has to bear that in mind. Okay, I'll stop there. Thank you.